Assalamualaikum and hello everyone. The objective of practical 1, basic concept of statistic and variation are to calculate and describe the following descriptive statistic: mean, variance, and standard deviation. Second, to understand the concept of correlation. And third, to determine the relationship between the two variables.
So, the methodology for the practical one is, first, we took 10 leaves from 3 different species. All the leaf samples were taken from all around the campus. And this is the leaf sample that we took from in front of the PTAR, which is the library. We also took the leaf sample in front of the garden at the administration building. So here is the 3 samples of the leaf that we got from all around the campus. To record the data, the width and the length of leaf was measured in the lab. And the data was recorded in the table. We also weigh the leaf to see if there is different weight in each of the sample that we get. And this is the leaf for the set 1, and this is for the set 2, and this one is for the set 3. Here is our result that we get from this experiment. Next, let us take a look at practical 2.
In this practical activity, we use a simple concept called quadrate to observe and analyze the difference in plant distribution present in a specific study site. The objectives of this experiment are to introduce the simple concept of quadrate, to enhance the power of observation, to record the difference in plant distribution within the quadrate, and to infer from natural observation the possible factors that affect distribution and abundance of organisms.
So, for the method for this experiment, a quadrate of 5 meter times 5 meter was made. Multiple quadrate samples were performed throughout the habitat at several random locations. All 3 stands that are found inside the quadrate were observed and the quantities were written down. The data was used to estimate the population size and population density within the habitat. So here is the sample for the quadrate 1 when we were taken in front of the K shop. It was really hot on that day, and that place in front of the cake shop, it's quite hard to stick a wood to make sure that the rope is not move around, because the saw at there is really hard to stick the wood inside it. So we need to hold all the rope at the same time, and then one of us take picture from above. After we ensure that our quadrant is not move around, we start to count how many species in that quadrant to record in our data table. Next, for the quadrant shoe, we took the sample in front of the administration building, which is behind the car park. And here is our result. For the quadrant 1, there is 3 species in the quadrant, and for the quadrant 2, there is 5 species in it.
Today, I would like to present the discussion of practical 2, which is sampling technique. Question 1. Define the term quadrant in practice. Quadrant is some random locations where in practice you plot the area that has been chosen with quadrant that has been set, which is by 5 meter times 5 meter at specific area. Within the quadrant should have all trees in several types of organisms to observe the species name, the total number of organisms that have been found in the area. The method depends on linear measurements between random points and adjacent individuals and between adjacent pairs of individuals. A quadrant is used in practice to mark out a specific area of the plant communities to be sampled. Next, question 2. What are the 2 basic requirements of all the quadrant techniques? Number 1. The area of the quadrant counted must be known so that density can be calculated directly. Number 2. The organisms must remain motionless during the counting period to ensure that they are counted and do not go missing. Question 3. What would be the advantages and disadvantages of using a larger or smaller quadrant to survey the sedentary organism? For the advantages, it is inexpensive because a few materials are required. It is a feasible and simple method to use when compared to other sampling methods. For the disadvantages, larger quadrats can provide more accurate estimates of overall densities, but they are more difficult to implement due to the time required to place and sample them. Despite the ease with which the quadrant can be designed, errors in studying may occur. Question 4. Explain the issues concerned by ecologists in conducting sampling method. Consider the sampling method that can be used to collect necessary data, methods for selecting sample locations to limit any potential bias and obtain the best estimates, methods for reducing the number of samples taken while still providing an accurate estimate of parameters. For the last question, question 5, discuss the factors that would have caused the difference in the number of plant species in quadrant. From the result, the Chronopteris linearis species dominated in both side 2 and 3. The plant is well adapted for Malaysia's climate, which is tropical and warm subtropical environments. Several precautions were taken. Quadrant is placed in an area with wide variety of plant. In conclusion, the objective of the experiment is to record the difference in plant distribution using the simple concept of quadrant while observing the factors affecting the distribution of the plant and enhancing the observation power. The data obtained was then used to predict the true population of the area.
As for the last experiment for these subjects, we did a field trip to Hutan Lipo Lenggeng, Negeri Sembilan to observe and relate the environment with what we had learned in chapter 9 and 10, which is community ecology. So, for the methodology for this field trip, we observe and analyze the conducted area by looking at the nature and relate them with the ecosystem there. Like how they do the habitat here, why they live together, is this type of environment is suitable for them. So, let us take a look at our result. First, this is Phylletis argina. It has thin crystallus that is white, grey and green-grey in color. Symbiosis existing here is commensalism because this type of lichen only use tree as structural perches but not extracting any nutrients or water from them.
Next is Xanthoria elegans, a type of lichen recognized by its bright orange and red pigmentation, has commensalism relation with rock but mutualism relation with fungi.
This is Naim hornum, or known as swan's neck-time moss. It is non-symbiotic organism because this type of moss obtains nutrients directly from its environment, so it does not depend on others. It also does not struggle for resource competition.
And this is Algomorpha quercifolia, or commonly known as oat-leaf fern. Competition happens between these species and the host plants as it competes for sunlight, nutrient, and water.
On this tree, this is the Phytophthora heye. It forms competition relationship with host plant to get more sunlight for photosynthesis. It may obtain nutrient indirectly from plant, but it is not parasitic as host plant may get nutrient from leaf litter around the host from it.
This is Physarum polycephalum, also known as the slime mole. It does not form a symbiotic relationship with other organisms as it interacts with food sources and microorganisms as biotic factors, while moisture, light and temperature serve as important abiotic factors.
Is Psilocybe pelliculosa. It forms a mutualistic symbiotic relationship with certain tree species as mycorrhizal association with the root of its host trees to absorb essential nutrients for host plant, while hosts provide carbohydrate and organic compound for mushroom.
Ganoderma lucidum, or the reishi mushroom, is a saprophytic fungus that breaks down dead organic matter, like decaying wood, to recycle nutrients and maintain ecosystem health. It plays a vital role in the nutrient cycle and overall balance.
Tramis pubescens is a saprophytic fungus that decomposes dead wood, recycling nutrients and contributing to ecosystem health. So it forms parasitic relationship with dead wood.
As you can see here, this is saprophytic fungus that colonizes dead or decaying wood, and it plays a vital role in the natural cycle of organic matter in the forest ecosystem. This is Ganoderma applinatum, the artist bracket or shelf fungus.
We also found tadpole in the stream nearby some rocks. Several environment factors that affect their species to be there depends on pH, salinity, predation, competition, water flow, and more.
When the ants have a symbiotic relationship with fungus, they cultivate fungal gardens, providing organic material as a nutrient source for the fungus, which in turn provide food for the ants. The ants protect the fungus and benefit from its nourishment, ensuring mutual survival. So as you can see here, it brings food from the tree to another place.
We also found spider web here, so we assume that there is spider habitat here. Spiders on leaves are influenced by abiotic factors like light, temperature, and humidity, which affect their survival. Biotic factors such as prey availability, predators, and competition.
After we were done our observation, we did community service at there. This is to ensure organism living there has a functioning ecosystem.
We collected rubbish such as paper, plastic, food wraps, some old ropes, and more.
We do hope everyone who is visiting there will keep this environment clean, as it is our responsibility to save our nature.
That's all from us. Thank you.